Good morning, everyone. Welcome to our COVID-19 webinar series brought to you by the Institute for Science and Policy, the Denver Museum of Nature and Science, and the Colorado School of Public Health. A huge thank you from all of us here and to our audience for joining us this Wednesday morning, slightly overcast here in Denver. I'm Kristen Uhlenbrock, I'm your host, and I'm thrilled for you all to join us today, where we're going to be talking about variants and mutations and the impact on vaccines. We actually have a little bit of a poll to kick us off today that we want to share with you. So Nicole's going to go ahead and launch that. And we're just really curious. We've been talking a lot about vaccines, and we want to see, of our almost 300 guests who've joined us this morning here, what's your vaccine status? Have you been fully vaccinated? Do you have one dose or more already in your arm? Are you eligible and on a waiting list? Fingers crossed, you're eager to get one when it's your turn. You're unsure if you'll get one, or none of that applies to you. Really would love to see what that response is. We'll give you just a few seconds here. Great. Go ahead and maybe, Nicole, why don't we go ahead and close that and see what the results are? We have 20% of our audience today is already fully vaccinated with 2 doses, and then another 26% have at least 1 dose in them. And I think many of us here in Colorado have been following the latest announcements, and I think we went to phase 1B of the vaccine, which actually launched on Monday, which makes it more accessible to those 65 and older, as well as teachers and others. So great to see that many of those joining us today have got their vaccine, or they're eager to get on the waiting list. So today, like Nicole said in our intro, is definitely a topic that's front and center to probably many of our minds, which is the idea of variants and mutations and that impact on vaccines. The variants of SARS-CoV-2 are really emerging around the globe. We're seeing them everywhere, including here in the US. And scientists and public health officials are really trying to understand how dangerous these might be and what do they mean for our current guidelines that we have and our vaccine rollout strategy. So today we're going to learn more about what we know about these vaccines or about these variants and these mutations. What are they and how do they come about? And then how do they impact our vaccines and our immunity? And of course, hopefully, how do we want to stay one step ahead as this really rapidly emerging situation unfolds? We are joined today by a lovely guest, Dr. Angela Rasmussen. Dr. Rasmussen is a virologist at Georgetown University's Center for Global Health, Science, and Security. She is particularly interested in viruses that are highly pathogenic, newly emergent, or likely to emerge because of climate change, land development, and ecological disruption. She's currently focused on SARS-CoV-2 as well as other emerging pathogens with the potential to profoundly impact our global public health. She is frequently in the news, so you may have already seen and heard from her. She's all over both primetime news, and I've heard her on Science Friday and NPR, and any news story almost from the Washington Post and New York Times has got her on the phone and quoted her. She's an amazing science communicator, in my opinion, so I think we're in for a real treat today. Good morning, Dr. Rasmussen. How are you?
I'm great, and thank you so much for that kind introduction.
We're so happy to have you here. So audience, we're going to have a few slides and presentation for Dr. Rasmussen this morning, and then we're going to get into Q&A in the latter half of the program like we always do. If you submitted some questions in advance, I've already got this kind of queued up. And of course, please feel free, if you've joined us here on the Zoom platform, to open that chat and dump your questions in there, and we'll try to incorporate some of those into the discussion as we get later on. But let me turn the screen and the mic over to Dr. Rasmussen for a little presentation.
Great. So this is going to be fairly short presentation. Now let's see. Oh, I'm trying to share. Sorry, you'd think I'd have the hang of this by now. Okay, so I'm just going to give a very short presentation to kind of introduce you to the concept of variants and mutations and what are they, and is this abnormal? Because I think a lot of times when people hear the word mutation, they think of something like this, or they might think of cancer. But usually it's in the context of something that is sort of extraordinary or out of the ordinary anyways, such as the X-Men, that mutation always confers some kind of superpower But in reality, for viruses, especially RNA viruses, like coronaviruses, mutation is actually a completely normal part of the process. So before I get started, though, I always start my talks with the territorial acknowledgement and equity statement. Today I'm in Seattle, and I'm presenting from the unceded ancestral homelands of the Duwamish people. And I acknowledge and honor the first people of these territories and their tribal governments, their histories and ancestry, and their roles today in caring for these lands. And I'd also like to acknowledge the history of systemic inequity in academic science that has really spanned centuries. My prior institution until November of last year, Columbia University, and my current institution, Georgetown University, were founded using profits from the transatlantic slave trade and the sale of enslaved people. And in addition to that, they excluded women and people of color from the academic community for more than 200 years. This has left a long and painful legacy of racial and gender-based inequality that continues to this day. So I'm encouraging everybody watching this today to consider how you can contribute to making scientific research a more equitable enterprise. And with that, I'm going to get started with this very quick presentation to just describe what mutation and virus evolution is and what we should be thinking about in the context of these variants. So on the left here is a panel showing mutation rate relative to genome size. And SARS coronavirus 2 is an RNA virus. Its genome size is right here in the range of 1,000 to tens of thousands of base pairs. In particular, SARS coronavirus 2 has a genome that's 30,000 nucleotides long. So that's GATC, GATC, sorry, it's early here. In the case of RNA viruses, it's actually GAUC, but the genetic code, that's what we refer to when we're talking about genome size. We are higher eukaryotes, and so our mutation rate, as you can see, is much lower than it is for RNA viruses. So they mutate all the time. And what does this look like? So here's just an example of a parental virus, and I've shown a little coronavirus here. And it's going to do what viruses do. It's going to get into a host and it's going to replicate. When it replicates, it will copy its genome and it will make mistakes some of those times. And those mistakes are called mutations. Those mutations occur randomly. So you end up with what are called quasi-species, a population of viruses that all have different individual mutations. And some of these mutations really do nothing. They won't be selected for in terms of evolution. Some of them will be in a place that is actually harmful to the virus. This is what we call a defective interfering particle. Basically, it's a viral particle that has a genome inside that's defective in some way. And then we might also have some mutants that confer some type of advantage to the virus. As these viruses go out and repeat the cycle of viral replication in a new host, eventually these mutants that have an advantage will take over the population. So you end up seeing a lot of these different mutants that have this blue genome that's advantageous to the virus compared to the parental virus. And of course, the mutants with defective genomes don't replicate and they're not preserved. So those are what are under what's called negative evolutionary selection. This would be under what's called positive evolutionary selection. So we've all heard about these variants of concern, and people seem to think sometimes that there's only these new variants and that all the other SARS coronavirus 2 strains that have been circulating are the same, and that's not true at all. This is a phylogeny from nextstrain.org, which analyzes and compiles all the different SARS coronavirus 2 genomes that have been sequenced. And as you can see here, there are many different variants that have emerged over the course of the pandemic. If you go onto Nextstrain and you look at this phylogenetic tree, you can mouse over these individual dots and they'll tell you exactly what's different about this from the original parental virus that was collected and isolated a long time ago. It feels like a long time ago, over a year ago in Wuhan, China. All of these are different and they are different evolutionary lineages that are related to each other. So at some point, for example, this lineage diverged and these variants emerged from that branch of it, these variants emerged from that branch of it. So there are many variants, but why are we worried about some of these other ones? And that's what I'm gonna talk about next. This is also from Nextstrain though, just really quickly. This is to show you how there's different global distributions of these different variants. Oops, And just to point out, the ones that we're worried about are these 501Y, V1, V2, and V3. This is the one that was discovered in the UK. This is the one that was discovered in South Africa. And this is the one that was discovered in Brazil. And if you look at those countries, you can see there's a big chunk of this pie here that is the P1 variant from Brazil. In the UK here, a big chunk of it is the B117 variant. And in South Africa, a very big chunk of it is the B1351 variant. So let's talk a little bit more about specifically what mutations these variants have. This is a schematic of the SARS coronavirus 2 genome. This is how the virus is organized. In terms of what the virus particles look like, it is an enveloped RNA virus in which the RNA genome, which is single-stranded and positive sense, is complexed with the N protein or the nucleocapsid protein, and it's stored inside this shell that's bounded by a lipid envelope. In that envelope is the E envelope protein, the M matrix protein, and of course the spike protein that the vaccines are designed towards. When this gets into a cell, the genome will go into the cytoplasm or the inside of that cell, and the genome will be translated into a polyprotein that's encoded by this ORF1A and ORF1B, sometimes called ORF1AB, because we like to make names difficult and confusing. There are also these subgenomic RNAs. So these are transcribed and then translated after the virus gets into the cytoplasm during replication. So there's individual RNAs for all of these. This includes the spike protein, the E and M protein, and the N protein. These are the structural proteins, as well as all of these other accessory proteins, some of which we kind of know what they do and some of which we don't. But that is where the mutations go. They go into all of these different proteins or genes encoding proteins, and they can have an impact on what the virus is actually doing, particularly mutations in these structural proteins, such as the S protein, as we'll probably talk about later. So these are the three different variants of concern. I'm a mess with my mouse this morning, I apologize. And these are just all of the individual mutations they have laid out in what genes they are in. And you can see here that of interest to everybody are these mutations in the spike protein. Now all three of these variants have this N501Y mutation, which is in what's called the receptor binding domain of the spike protein. And the receptor binding domain is the part of the spike protein that interacts with ACE2, which is the virus's host cellular receptor. So when spike interacts with ACE2, that essentially is the signal. It's like a lock and key that the virus needs to enter and infect the cell. So these mutations are thought to be very important, both in terms of the virus being able to infect cells, as well as antibodies, since antibodies directed at the receptor binding domain are thought to be very important for neutralization. You can see here too that there are a variety of other mutations that are also in the spike protein. Some of them are shared between these variants and some of them are not. We're still really looking into what they're all doing. And that kind of gets to my last slide here. So we know that these three variants are of concern because they're more transmissible, but we don't know how they're more transmissible. So all of these mutations, it's great to know what they are. It's great to know where they are in the genome, but that's a different story than actually knowing what they're doing to contribute to the increased transmissibility that's been observed. So there's a couple different possible mechanisms of increased transmissibility, and that's what people are working on right now to figure this out. One possibility is that these mutations increase viral fitness, which means they make the virus able to replicate better. This could lead to increased viral shedding, and you can imagine how that might make a person more contagious if they're shedding more virus from their nasal passages. It also could be a longer interval of contagiousness, meaning that a person is contagious for a longer period of time than they would be if they were infected with one of the quote unquote old variants. Another possibility is that they could increase receptor binding affinity. And we already know that the N501Y mutation that I just mentioned does in fact increase receptor binding affinity to ACE2. So that could cause increased infectivity, meaning that you don't need to be exposed to as many virus particles to be infected. That means that it's basically the virus is more efficient at infecting cells that it otherwise would normally be. And we know that not only does N501Y increase binding affinity to ACE2, but some of the other mutations that have been found specifically in the B1351 and P1 variants, first discovered in South Africa and Brazil respectively, have other mutations that amplify this effect, so that they're capable of binding even more tightly to ACE2. And then another possibility is that this could actually increase virus particle stability, or virion stability. That's what we call a virus particle. Mutations in all of these structural proteins that I was just mentioning, S, E, M, and nucleocapsid, could make the virus capable of being stable in the environment for a longer period of time. So if you're in an enclosed space with somebody who's infected with one of these variants, maybe you don't need to be in there for as long to be exposed to a sufficient dose of infectious virus to become actually infected. And that's the end of my slideshow. And I'm looking forward to talking about all of this with you. And I hope that that was helpful in terms of just laying out some of the basics about what these variants are and what we think they might be doing.
Yes, thank you. We really appreciate that level of detail too. And I want to try to unpack some of that. We've got a lot of questions already coming in. And then I think we want to also move into the vaccine discussion around these and what we know or don't know about that. So I'm curious, from the mutation pathways of these viruses, do we know if that mutation pathway would turn it into a kind of less effective or more contagious virus? What's known about that?
So we don't, and that's one of the troubles with working with a virus that is brand new. We know, of course, that the spike protein is important. So any type of deletion of the entire spike protein of the receptor binding domain is not going to result in an infectious virus. But individual point mutations, like what I was just showing, or individual very short deletions, a couple of these mutations are actually deletions of 1 or 2 nucleotides. We don't really know what kind of effect that will have. The best we can do is compare it to other related coronaviruses that we have studied in more detail, such as SARS coronavirus classic. The problem with that is that SARS coronavirus 2 has some features genetically that SARS coronavirus classic does not have. And obviously it's a different virus that causes a very different disease. One of the reasons why COVID-19 has become a pandemic and SARS classic did not is that it can be transmitted pre-symptomatically. With SARS coronavirus classic, if you had SARS, that's when you became contagious. So if you were getting a fever, if you were developing respiratory illness and became symptomatic, you could be isolated without requiring a test. With SARS coronavirus 2, you can be transmitting virus without having any idea that you're actually sick. So there are a lot of big differences between both the viruses, and even though they're closely related, and the diseases that they cause, which makes it very, very difficult to just use SARS and what we know about that to make conclusions about what's functionally important for SARS coronavirus 2. Unfortunately, the way that we figure this out is by basically making mutant viruses, infecting cells and animal models with them, and seeing what mutations have an effect and which don't. And to further complicate this, sometimes there's a phenomenon called epistasis, in which multiple mutations will have a synergistic effect. So you have to get them in the right combination when you test them to actually see that effect in the lab. So it really is very challenging, and that's really why we can't just look at the sequence and say, aha, N501Y, we know that that's important, because many parts of the genome, we just don't know what's functionally relevant and what's not for the virus or for the host.
There's a great question here from our participant, Melissa, that asks, if there are so many of these mutations, and the 3 that you highlighted, what makes them particularly noteworthy? What do we know about them? Are they causing more severity in the disease? Why are these 3 that we're currently focused on, at least for this presentation and globally, of note?
So there has been some very, very limited data from the UK, and I'm pretty skeptical of it, actually, that says that the B117 variant actually increases disease severity or increases mortality risk. I'm I'm not sure about that, just because that's based on a pretty small subset of patients that they analyze to determine that. But the real thing that all three of these variants have in common is that they're more transmissible. And the reason why this was even on anybody's radar is that these variants became the predominant circulating variant in the regions where they emerged very, very quickly. So by contrast, there's another mutation that was characterized back in May called D614G. That's a change from an aspartic acid to glycine. I'm sorry, I'm still not caffeinated enough yet this morning, in the spike protein. And because that was becoming dominant in the US and in Europe, people suggested that it was more transmissible. And honestly, there may be a slight transmission advantage after months of doing experiments on that particular variant. But it's not as clear as that. And it took months to become the dominant circulating strain. The B117 variant, for example, became dominant in 1.5 months in Southeast England. So that's unusual. That suggests that the virus is able to outcompete effectively all the other viruses that are circulating around there. So we know that all of these are more transmissible. And the reason why that's a concern is that with COVID, really the hospitalizations and deaths are kind of a numbers game. The vast majority of people who get COVID will not end up going to the hospital because of it. And as many people who want to downplay the severity of the pandemic will point out, there is a fairly low case fatality rate of SARS coronavirus 2. But if you get enough people infected with it, then you are going to start seeing those numbers skyrocket, which we've actually seen here without these variants. And then you'll see a coincident burden on the healthcare system. That can lead to other morbidity and mortality because healthcare resources are strained or used up and healthcare rationing has to be implemented. So that the increased transmissibility and the potential for additional surges is really what's of concern with regard to these variants, much more so than increased virulence.
Great, thank you. How difficult is it to detect these emerging variants or variants that are there? Like, how good are we at detecting them?
So the UK is very good at detecting them, which is why they detected B117. They sequence about 10% of all diagnosed SARS coronavirus 2 cases. In the US, not so much, which is why it took us a little longer to figure out that these variants were actually already here. Because we were sequencing less than 0.5% of all of the sequences of all the SARS-2 positive cases that came in. Genomic surveillance is really, really important. And I'm glad to see that now the CDC is under leadership that recognizes the importance of that. And there is starting to be more dedicated funding for that. But prior to this sort of wake-up call about these variants, the genomic surveillance that was occurring in the US was mostly people using their own grant funds, their own research funds to do a little sequencing here and there. There are a couple surveillance programs that were for influenza that have been somewhat repurposed, but we're not doing nearly enough genomic surveillance. So I'm glad to see that at least we're learning that lesson, that we need to be doing more of that.
Great, thank you. So there's some research and some talk about being reinfected from COVID-19. Is that due to a new strain? What do you know about reinfection rates, and how does that tie in or at all to variants and mutations? And then I want to move into a little bit about the antibodies and vaccines after this, but what do you know about reinfection and what does that mean for variants?
Yeah, so this is a pretty good segue actually into antibodies and vaccines, because the short answer is we still don't know that much about reinfection. We think that in general, reinfection is fairly rare. There would be epidemiological indications if people were getting COVID and then months later coming back and being put in the hospital again with COVID. And that's not happening all the time, but it has happened. So we know that reinfection does occur, and this reinfection has been confirmed in some of these cases with sequencing, confirming that they were actually infected with a different variant of SARS coronavirus 2. And people have been very alarmed at the finding that in South Africa, where there were a high percentage of people participating in a clinical trial who previously had COVID-19, then tested positive, suggesting that they were reinfected with the B.1.351 variant at a rate of close to 4%. Now that's alarming, but there's also no indication, we don't know that much, first of all, about what those people's immune status was. We don't know if maybe some of them had lower levels of antibodies, if they didn't mount good immune responses to their first infection. We don't know that that's going to be the case with vaccines, because this rate of reinfection was reported in the placebo group. So that, there's no way to tell how vaccine-induced immunity would protect against that variant. Also, those people, there's no indication that they were very sick from their reinfections. So that may suggest that while the variant is capable of evading some immune responses, it's not capable of evading all of them. And people who've previously been infected are still protected against severe disease, even though they may be susceptible to reinfection. But it's an important area that there's just a lot of knowledge gaps in right now. So we definitely need to look into it more. But I'm not as worried about it as some of the headlines have suggested we should be, just because, those people, it doesn't seem like all those reinfection cases were ending up in the hospital with their reinfection case of COVID.
So there's also been news reporting, and South Africa has been making a lot of news with the AstraZeneca vaccine and the other vaccines there. But the UK has also talked about spreading out the dosages of vaccines to try to get more people at least a first shot. What's your thoughts on that as a strategy for vaccine implementation?
So I'm opposed to that strategy without data to support it. With the AstraZeneca vaccine, there is data to support it. There's data that suggests that spacing out the shots by up to 12 weeks, longer intervals actually can result in more protection. So for that vaccine, sure, go ahead. Like 12 weeks is great for the second shot. For the Pfizer vaccine, there's just no data to support that. And I know that in the UK, they've said you can do 12 weeks between shots for both AstraZeneca and Pfizer. Those are two very different vaccine platforms. The Pfizer vaccine is an mRNA vaccine. The AstraZeneca vaccine is an adenovirus vectored vaccine. They work pretty differently in terms of how they express SARS coronavirus 2. I'd be very reluctant personally to suggest that anybody getting the Pfizer or Moderna vaccines space it out beyond 6 weeks, which is the limit to which those dosing intervals have been tested. There is some data suggesting that you can go up to 42 days after your first shot and still have equivalent protection for Moderna and Pfizer. But 12 weeks, I'm not sure. And the reason I think that could be dangerous, because a lot of people are like, well, we have other vaccines and we sometimes wait years before giving a booster shot for those. What's the harm? The harm is not that I think that a second shot given 12 weeks after the first shot would mean that the vaccine wouldn't work. It's that people might not be protected for that entire period of time from the first shot alone. And there is some data that suggests that for both of Moderna and Pfizer vaccines, you really do need that second shot to induce high levels of neutralizing antibodies, which are the antibodies that will render the virus non-infectious. So for that reason, without any data suggesting otherwise, I recommend that people get their second shot on time, at least within 6 weeks of the first shot.
So is there anyone working on a vaccine that is potentially going to protect against all these variants, right? And is there such a possibility as a kind of a pan vaccine? We had antiasis, and a couple other people have been asking about this too.
Yeah, I think in the future, certainly there is the possibility of developing a more universal COVID vaccine. The reason we can't do that right now is that we don't know what mutations are going to be important or advantageous for the virus. So it's really hard for us to predict how the virus is going to change, and specifically how the spike protein is going to change. So the spike protein is what is called the antigen. That's the target that antibodies are elicited to during vaccination. And we just don't know how the antigen is going to change. And if we have a little more information as the pandemic goes on, as we start to control this, we will be able to see, the variants that are going to emerge will emerge, and then we can develop boosters and potentially a universal vaccine. I think that it's very wise, even though there's no evidence that the vaccines for the most part are completely non-protective against these variants. There may be reduced efficacy for some of them, but it's not completely shot. So I think though that it is wise to start making boosters. And the good thing about these vaccines is that it is actually very technically easy to change them, especially the mRNA vaccines. It's just a matter of, if you think of mRNA as a message, it's a matter of changing the spelling of a few words effectively. So there is a more complicated regulatory process that they'll have to go through, and I'm not sure how that is going to work. But we do this every year for influenza vaccines. We change them based on what strains are circulating. So I'm confident that we can do this as well for these SARS coronavirus 2 vaccines. And it may mean for most people realistically that you're just getting a booster.
So are there potential ways that are being explored in the research of the biotech besides vaccines to change the course of viruses and how they interact with our DNA?
Well, so RNA viruses don't really interact with our DNA. And maybe I should have explained this a little better in the beginning. When the RNA virus genomes go into the cytoplasm, that's the inside of the cell. That's not the total inside of the cell. The inside of the cell where our DNA is the nucleus of the cell. And most RNA viruses, not all, but most, including coronaviruses, don't get in there. mRNA doesn't get in there either. So mRNA is generated in the nucleus when it's our mRNA, and then it's exported out where it gets translated into protein. The mRNA vaccines never go into the nucleus either, so they can't really interact with our DNA. Going forward, though, I mean, this is a really broad, interesting question that I could probably spend the entire time talking about, because this is actually what I study, and that's the host response to viral infection. Viruses have to interact with our cells and our proteins in those cells in many different ways. And it's actually not well understood for any virus. Some of those interactions are well understood, but not all. So the virus, when we say the viruses hijack cells to get into them and to replicate, that basically means that they're appropriating different types of cellular machinery to replicate themselves. They're also usually messing with the host immune system, including the innate cellular immune system, so that the cell basically isn't trying to defend itself against them. So there's all these different interactions, and they're really important in terms of pathogenesis as well as susceptibility and long-term immune protection. But I mean, that, like I said, that's my entire area of research. I could spend all day talking about this, so I might just leave it at that, because otherwise I want to make sure I get to all the questions.
Yeah, that's great. No, I appreciate just that kind of quick snapshot of that. And we are not going to get to all these questions. We have wonderful questions coming in right now. So I want to take one from Jenny. We're asking, what is the easiest way to get an antibody test? And then how quick are the results available? And maybe I should ask too about accuracy. I know, there's some, I don't know how accurate antibody tests currently are.
So that depends on the antibody test for all of the above. But you should be able to get an antibody test just by asking your provider to order one for you. The question is, or you can go to an urgent care clinic. I know in New York, they were offering antibody tests last summer to people who wanted to come there and see if they had gotten COVID during the spring surge of last year, when PCR tests were very unavailable for people, and people didn't know if they'd had COVID or not. So you should be able to ask or request an antibody test. The problem there is there are a lot of different antibody tests, and not all of them are created equal. So you're going to want to ask your provider, when they're ordering that test for you, to order one that is both highly sensitive and highly specific. And that should be in the package insert. The major manufacturers of antibody tests are pretty good from what I've seen. And it also, one thing to think about too is what you want the antibody test for. If you want to see that you're having a good response to the vaccine, you also need to ask your provider to make sure they're ordering an antibody test that's looking specifically for anti-S antibodies, so antibodies against the spike protein. Some of the antibody tests look for anti-N, And that's actually really good because you can distinguish in a vaccinated person, for example, if they've actually been exposed or infected with coronavirus after vaccination. If they haven't, they won't have antibodies to N. If they have, they will, because the vaccines are only directed at the spike protein. So people who have never had COVID who are vaccinated will only have antibodies against S. People who've been infected will have antibodies against both S and N and any other antigen. So if you want to just see if you had COVID in the past, any antibody test will do. If you want to see that you're having specifically a good reaction to the vaccines, then you want to make sure that you're asking for an antibody test that specifically looks at antibodies to the spike protein.
Thank you. How is our interconnected nature shaping this pandemic, both currently from our response and hopefully the eventual control? As some have put it, potentially this could be like the seasonal cold or flu season for us. What does that look like from your perspective?
This is a really hard question to answer. And a lot of people have asked me questions along this line, like, another version of this question is, do you think the virus is going to become endemic? I think that there's a good chance that it will. I think, though, it's very difficult to forecast because of a couple different things. One is, how quickly can we vaccinate as many people as possible? Another is, pardon me, another is how many people are willing to actually be vaccinated once we do have those supplies. A third is, are people going to continue to take precautions right now in the short term to reduce community transmission? And a lot of people have been discussing this as we need to get vaccines out faster, faster, faster. And we do. But right now, while we're still having supply shortages, we also need to really double down on our efforts to reduce transmission in the community. That means increased adherence to masking, physical distancing, ventilation if possible, avoiding gatherings in enclosed spaces, hand washing and disinfecting high touch surfaces, staying home if possible, acknowledging that this is not possible for everybody. But the more people can apply any of these measures whenever they are interacting with other people in the world, the more they're going to reduce their own exposure risk, the more they're going to bring community transmission down. If we can bring community transmission down while we simultaneously ramp up vaccination, I think that we have a very good chance of really getting this under control sooner rather than later. Now that doesn't mean that it won't become endemic, because the larger problem there is that as we go back to international travel and as we sort of go back to quote unquote normal life, we're going to have to contend with the fact that even if we get, to herd immunity in the US, there's not going to be that globally. And we need to really think about how we're rolling out vaccines globally in an equitable way, because until all of us are safe, none of us are safe. So I really, when people ask me that question, I mean, my short answer is, I don't know. If you do this, it'll be sooner for us in the US. I think that I would really encourage people to think about, the long-term end game of this. This is not going to be something that's going to just go away overnight. The end of this pandemic is going to be more gradual. And, I know that this is tough. Like this sucks. I hate it. I hate being stuck in my house. Like, I love doing stuff like this, but I hate the fact that this is like, a lot of my interaction with the real world and with people in other places. Normally I'd go to a conference in person and give a lecture like this. I hate having to do this over Zoom, and I'm tired of it, just like everybody else is. But I really do encourage people to just hang in there a little bit longer. As we get more and more vaccines out to people, we will be able to start sort of easing back into a more, again, quote unquote, normal life.
I appreciate that. And you're a science communicator too at heart, in addition to being this amazing virologist and researcher. We have a story, and I'm not going to read the whole story shared by one of our guests, but in essence, this person has had received 2 doses of the Moderna vaccine and had side effects, is a healthcare worker, and is of course very much encouraging others, to get the vaccine because of how it protects. But there is concern that these effects, could potentially impact compliance in other years or yearly shots. And I think I had read, there are some side effects, right, to the receiving the vaccine dosages. There's no deaths, right, associated with the kind of rollout of the vaccines. So just what are some of your thoughts on that, and any sort of like tips for communicating with people that we love and others that we care about why we should be getting vaccinated?
Yeah, absolutely. And that is a very legitimate concern, because many people have reported that the side effects, while again, not lethal, not permanent, are difficult for many people, and more difficult, for example, than like your annual flu shot. Some people have reported being, knocked out for 24 to 48 hours, especially after the second shot. And understandably, that makes people reluctant to take the vaccine. So what I've suggested are a couple things. And I suggested this to my parents, who got their first shot of Moderna a few weeks ago, and are getting next week their second shot. I asked them, how was the first shot? They said, oh, well, we had some muscle pain, but otherwise it wasn't too bad. But they've asked, they said, I heard it's a lot worse after the second shot. And for some people that has been true. I think that early on, or not early on, but when the phase 3 clinical trial started to read out, one thing that could have been done better was communicating with the public about those side effects up front. I think that it has been more difficult than it needed to be, because that wasn't really disclosed alongside the very encouraging and awesome top line efficacy data. And I think that it is really important to be honest and open with people about some of the things that might make this difficult, so people can prepare themselves. I think that nationwide, there should be some policy changes to support this as well. And this actually makes it easier for people to do non-pharmaceutical interventions too. For example, paid sick leave. If people could take a day or two off after getting the vaccine, plan ahead for it, I think, and not worry about their jobs and not worry about not getting paid, I think that would make it a lot easier for people. I think it's a tough ask to say that, okay, you might basically have flu-like symptoms where you feel like crap for a couple days after you get the vaccine, but it's worth it. But guess what? You still have to go to work. So I think there are things we could do to prepare people for it better and make it easier. And going forward in the future, if there is a need for boosters, we don't know anything about like if a third shot would be even worse in terms of the side effects. So that's definitely something that needs to be looked at. We also need to start thinking about doing what's called heterologous boosting. And that is when you use a different vaccine platform to boost. Now, I don't know if that would have any effect on side effects, but the side effects reported for the Novavax vaccine, for example, which is a protein subunit vaccine, different than mRNA, that's actually giving you the spike protein, has been associated with fewer side effects. And same with Johnson & Johnson. So it may be possible that if you can sort of mix and match booster shots, that we may be able to help people get away from some of these side effects. So I think that it's a combination of just being honest with people about the side effects. Also, there is one encouraging thing about the side effects, and that is that, and most people I think don't know this, but when you get sick with the flu or with a cold, the symptoms are largely the product of your body's immune system responding to that infection. And that's what the side effects are for this virus too. So if you do have bad side effects, it actually just means that your antiviral machinery is pumping out a ton of interferon, which is an important molecule for recruiting antiviral immune defenses. It means that the vaccine is working. And I think that does give people a little bit of comfort that, okay, I feel terrible, but it's a lot better than having COVID. And it's a sign that also this vaccine is eliciting robust immune responses and it's going to protect me. So I try to cover all those points when talking to people who are concerned about the side effects and what they might mean. And ultimately, to drive one more point home, having those side effects for 24 to 48 hours is much better than having COVID. Because even if you have mild COVID, you know, as many as 30% of people have reported having some symptoms of long COVID. It's something that doesn't necessarily go away. It's not transient. These side effects are. So get the vaccine, tough it out through the side effects if you can, and know that you're not going to have a much longer term problem because you're protected from COVID.
Thank you. That was a great response. We're coming up at the end of our episode, but I want to give you one final word if you have a thought, and just want to share, you know, what's your greatest hope for the rest of 2021?
So my greatest hope is that people actually just hang in there for just a little bit longer, long enough for us to increase the supplies of vaccines so that people can be vaccinated on demand. As I was saying earlier, I'm really concerned with the idea that just because we have the vaccines, that we're completely in the home stretch and we're free and clear. We're not, and these variants are just a real red flag about that. I think that if people can just hunker down for a couple more months, this is not eternal lockdowns, this is not eternal restriction. But if people can hunker down, hang in there for a couple more months, get community transmission down while we simultaneously get vaccination up, we will be able to go back to a more normal life. We will be able to go indoor dining. We will be able to go to bars. We'll be able to hug our parents. We'll be able to hug our kids and see our kids and grandkids. We'll be able to do all those things that we've been missing out on doing through a combination of both elimination through targeted interventions and vaccination. I worry, as much as I love vaccines, and vaccines are, you know, certainly the greatest public health triumph of the modern era, although I've argued with people as to whether sanitation and water are equally good. They're also good. But vaccines have prevented millions of deaths, hundreds of millions of deaths worldwide. Not just a COVID vaccine, all vaccines. Vaccines are just a triumph of public health. I love them, but they're not the only thing that's going to get us through this. And I really hope that these variants are a wake-up call for people, because if we don't get transmission down, new variants might also emerge. As I said at the very beginning of my talk, mutation is a normal thing that happens when viruses replicate. If you give the virus more opportunity to replicate, new variants will emerge. So we need to take away those opportunities for the virus to replicate while simultaneously protecting ourselves against it. And if we can just, again, for a little while longer, you know, find that extra burst of energy to make it across the finish line, I think that we're going to have a very nice summer, fall, winter 2021. So that's my greatest hope, is that people will, you know, stop looking at this as a sort of one-sided thing. Vaccines aren't the answer to this. Immunization is, but also the non-pharmaceutical interventions are a part of that bigger picture.
Thank you. What a joy to have you today. We are so grateful that you got up at 7:30 a.m. your time to spend this very in-depth discussion with us, and you handled all these questions beautifully. It was super rapid fire. So thank you for joining us today.
Oh, it's really, really my pleasure. Thank you so much for having me. I really, really love being able to talk to people directly and to answer their questions. And the questions are fantastic. And I just wish that I could get to more of them.
I wish that too. Every time we have a wonderful audience. So thank you to our audience for joining us today, for all your thoughtful questions and feedback. As you know, we're going to continue on with this series. So hopefully you'll join us back here on February 24th. So 2 Wednesdays from today. We'll be hosting Dr. Harvey Feinberg, current CEO and president of the Gordon and Betty Moore Foundation, former president, of course, of the U.S. Institute of Medicine, also known as the National Academy of Medicine. And we're going to start off with him in a 3-part mini series of this bigger series, where we're going to talk about what does that road to new normal look like, right? And start to put on a little bit of that thought process with some really exciting speakers in the mix. So hopefully you'll join us back here on February 24th. Thank you everyone for staying on for a few minutes longer with us. I did not want to end this so short. Again, thank you to Dr. Rasmussen. Thank you to our partners at the Colorado School of Public Health, health, and all my museum colleagues. I hope you all stay safe and well. And we do have a transcript if anyone asks, because there was a lot of information today. So not only do we have a video recording of this on the Institute YouTube channel and other social channels, we do have a written transcript that should be up in about 24 hours if you prefer to kind of read this discussion today. So thank you, everyone. Stay safe, stay healthy, and hopefully we'll see you back here in 2 weeks.